Selamat siang Bapak dan Ibu. Uh, Sambil menunggu Pak Direktur hadir di ruang Zoom kali ini, kita acaranya akan dimulai pukul 13.35 ya Bapak dan Ibu. Sambil menunggu Pak Zainal join ke room ini. Terima kasih. Baik Bapak dan Ibu, karena Pak Zainal sudah hadir bersama kita, acara segera kita akan mulai. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang Bapak dan Ibu sekalian. Salam sejahtera bagi kita semua.
Oh, sekalian hiko, tapi hikonya nggak murah dia jual.
Kenalkan, saya yang akan membantu acara pada siang hari ini. Saya Indra dari Kepustakaan BRIN Kawasan Jakarta Gatot Selamat datang dalam acara Sosialisasi SciFinder yang diselenggarakan oleh Direktorat Repositori Multimedia dan Penerbitan Ilmiah RMPI BRIN Rabu 15 November 2023 pukul 13.30 sampai 16.00 waktu Indonesia Barat melalui Zoom meeting. Yang terhormat Bapak Zainal Akbar, MCom PhD, selaku Direktur Repositori Multimedia dan Penerbitan Ilmiah, Ibu Nurika Retno Widuri, Esos MIR, Koordinator Layanan Kawasan Serpong, kemudian kepada Kepala Organisasi Riset dan Pusat Riset, serta para narasumber dan riset, serta para tamu undangan yang berbahagia. Agenda kita pada siang hari ini, kita akan mendengarkan mengenai mengenai platform SciFinder dalam riset yang akan dibawakan oleh Bapak Yudi Dwi Kurniawan, SSI MSc PhD, kemudian penggunaan SciFinder untuk riset BRIN, perwakilan dari CIS SciFinder. Sebelum ke acara utama, marilah kita dengarkan sambutan dari Direktur Repositori Multimedia dan Penerbitan Ilmiah, Bapak Zainal Akbar, MCom PhD. Kepada Pak Zainal, kami persilakan.
Oke, tes. Baik.
Ya, Pak.
Baik, terima kasih, Sindra. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Selamat siang, salam sejahtera buat kita semua. Terima kasih saya ucapkan kepada para narasumber. Oke, ya. Jadi terima kasih kepada Pak Yudi yang sudah menyempatkan waktunya untuk sharing gitu. Jadi kalau bagi saya sendiri, SciFinder itu sesuatu yang masih asing. Nah, kemarin itu diadakan itu karena memang ada permintaan dari teman-teman periset. Nah, harapannya dengan kegiatan ini kita terutama periset ini bisa tercerahkan, SciFinder itu bisa dimanfaatkan untuk kegiatan apa saja. Harapannya kami tentu saja dari direktorat nanti bahwa ini akan dimanfaatkan oleh teman-teman periset sehingga nanti kami bisa evaluasi utilitasnya sampai seberapa begitu. Dengan harapan tentu saja jangan sampai kita sudah melanggan kemudian ternyata pemanfaatannya masih kurang. Nah, ini mungkin seri pertama, mungkin saya yakin akan ada seri-seri berikutnya mungkin yang semacam ini, bagaimana platform SciFinder ini dimanfaatkan. Oke, mungkin itu dari saya, Mas Indra. Selamat mengikuti sosialisasi ini. Semoga ikhtiar kita untuk mengenal platform SciFinder ini secara khusus dan juga riset secara umum itu dapat kita capai dengan baik. Terima kasih. Saya kembalikan ke Mas Indra. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Pak Zainal. Baik, langsung saja kita menuju acara utama. Mari kita dengarkan pengantar mengenai platform SciFinder dalam riset yang akan dibawakan oleh Bapak Yudi Dwi Kurniawan, SSI MSc PhD. Kepada Bapak Yudi, kami persilakan.
Terima kasih banyak, Mas Indra. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, Bapak Ibu semuanya. Yang saya hormati Bapak Zainal Akbar, selaku Direktur eh, Repositori Multimedia dan Penerbitan Ilmiah. Yang saya hormati pula kepala organisasi riset dan pusat riset di lingkungan BRIN dan para sivitas yang hadir pada Zoom meeting kali ini. Perkenalkan, nama saya Yudi Dwi Kurniawan. Saya dari Pusat Riset Bahan Baku Obat dan Obat Tradisional BRIN. Pada kesempatan kali ini saya diminta untuk sharing pengalaman sebetulnya mengenai penggunaan platform SciFinder ini dalam menunjang riset yang dilakukan di BRIN. Pertama, mungkin masih ada dari Bapak Ibu sekalian yang masih asing ya, SciFinder itu sebetulnya apa gitu. Jadi mohon maaf, ini saya tidak menyiapkan PowerPoint karena mungkin lebih enak untuk sharing secara langsung ya. Karena ini biar mengalir saja dari pengalaman begitu. Jadi SciFinder itu sebetulnya sepemahaman saya adalah semacam search engine Bapak Ibu. Jadi kalau kita secara awamnya itu seperti Google gitu ya. Kalau kita mau cari sesuatu atau eh, Google Scholar mungkin lebih tepatnya ya untuk kita sebagai periset. Dan yang membedakan SciFinder dengan yang lain, platform yang lain itu adalah pertama, SciFinder sendiri sebetulnya didesain secara spesifik untuk kimia, untuk di bidang kimia. Namun, namun demikian, para pengguna yang dapat memanfaatkan platform ini tidak terbatas hanya pada periset yang berkecimpung di bidang kimia, namun di bidang-bidang lain yang berkaitan dengan eh, bidang kimia atau berkaitan dengan struktur molekul seperti itu, misal dari obat-obatan mungkin atau bahan alam atau material juga energi seperti itu. Jadi eh, yang pertama yang perlu kita garis bawahi adalah SciFinder ini didesain untuk kimia, namun penggunaannya lebih luas. Kemudian fitur apa yang membuat SciFinder ini jadi aduh, mohon maaf ini ada yang bersihkan AC ya. Mungkin apakah suara saya masih terdengar Bapak Ibu?
Terdengar Pak Yudi.
Terdengar ya. Oh baik, mudah-mudahan
Ya.
tidak mengganggu ya. Jadi saya lanjutkan Bapak Ibu. Fitur apa yang menjadikan SciFinder ini khas, spesifik dan bisa jadi menjadi salah satu fitur andalan yang banyak diperlukan oleh periset kita. Itu fitur yang ditawarkan berupa pencarian melalui struktur kimia, Bapak Ibu. Jadi kalau kita mencari literatur itu umumnya kan kita menggunakan keyword ya, berupa keyword gitu, berupa kata-kata, misal COVID-19 dan semacamnya. Sementara di bidang-bidang tertentu seperti kimia yang informasi pentingnya itu bukan berupa kata-kata, tapi berupa struktur kimia, ini kita masih masih punya tanda tanya ya, bagaimana caranya kita mencari sumber di literatur yang tepat sasaran, yang kita tidak untung-untungan, kadang dapat, kadang tidak, dan bisa terhubung ke sumber literatur yang terupdate. Itu disinilah peran SciFinder untuk menjawab pertanyaan-pertanyaan tersebut. Jadi SciFinder ini adalah search engine yang kita bisa gunakan untuk mencari literatur seperti biasa juga berdasarkan keyword seperti itu, juga berdasarkan struktur kimia. Dan yang menjadi poin apa ya? Poin utama dari platform SciFinder ini dia terhubung ke sumber-sumber jurnal yang ada di seluruh dunia. Itu yang saya pahami selama ini. Nah, pertama, lalu hubungannya dengan kita, seberapa penting SciFinder ini bisa kita gunakan, atau seberapa penting SciFinder ini bisa menunjang riset kita. Ini saya kasih bayangan seperti ini Bapak Ibu. Saya ambil salah satu contoh ya, salah satu aktivitas riset yang dilakukan di BRIN ini. Oh baik, mohon maaf. Saya sudah naikkan ya.
Terdengar kok Pak.
Oh terdengar. Oh
Dengar,
ya ya.
dengar.
Oke. Jadi kita coba lihat dari negara kita sendiri ya Bapak Ibu. Indonesia ini dikenal sebagai negara yang memiliki mega biodiversitas. Kita memiliki sumber kekayaan alam yang sangat melimpah yang belum semuanya itu dieksplorasi dan dimanfaatkan sebagaimana mestinya untuk kemaslahatan masyarakat. Seperti itu. Salah satu yang kita miliki itu kekayaan sumber daya alam seperti tanamannya, ada binatangnya, juga yang sumber daya yang ada di laut, makhluk hidupnya beserta lingkungannya. Kemudian juga kandungan yang terdapat di dalam sumber-sumber tadi seperti senyawa kimianya itu belum dimanfaatkan secara optimal. Jadi salah satu kegiatan riset yang dilakukan di BRIN ini adalah ini ya Bapak Ibu ya, terutama di organisasi riset kesehatan itu adalah pengembangan obat. Sebagai informasi, sepemahaman saya sejauh ini proses untuk menemukan obat itu melibatkan proses-proses yang meliputi pertama eksplorasi ya Bapak Ibu. Pertama eksplorasi sumber daya alam, ya kita bisa istilahnya mendapatkan informasi dari mungkin dari masyarakat atau memang dari tanaman yang sebelumnya belum pernah dieksplorasi atau dari sumber lain, kita ambil ekstraknya kemudian kita ujikan ke target tertentu. Kemudian ketika kita sudah mendapatkan ekstrak yang aktif, tahap selanjutnya kita melakukan pengejaran, identifikasi struktur senyawa atau senyawa major apa yang ada di ekstrak tersebut yang bertanggung jawab terhadap aktivitas tersebut. Kemudian setelah dapat senyawa tunggal yang menjadi komponen utama pada ekstrak tersebut, dilanjutkan dengan peningkatan efektivitas dari senyawa tersebut, juga penurunan efek sampingnya melalui modifikasi struktur kimia. Kemudian juga dilakukan pengujian secara in vitro, in vivo, sampai akhirnya bisa sampai ke masyarakat. Yang menjadi pertanyaan lebih spesifik, ketika kita melakukan eksplorasi untuk menemukan obat yang melibatkan aktivitas-aktivitas tersebut, kita melakukan pengejaran atau pencarian senyawa-senyawa yang ada di ekstrak tersebut. Pertanyaannya adalah ketika kita berhasil, misal kita berhasil untuk mendapatkan satu senyawa yang bertanggung jawab terhadap aktivitas misalnya antaria. Pertanyaannya adalah, dari mana kita bisa tahu kalau senyawa itu baru atau tidak? Karena informasi yang kita punya itu setelah kita kumpulkan data ya, untuk memperoleh struktur kimia dari senyawa yang kita peroleh itu bagaimana, hasil akhir yang kita dapatkan adalah struktur kimia dari senyawa. Kemudian kita mau cari di literatur, apakah senyawa ini baru atau tidak? Kemudian apakah kalau memang baru, kalau memang ternyata tidak baru, apa hubungannya dengan senyawa yang sudah ada? Itu sepengalaman saya ketika mencari literatur yang terkait langsung dengan struktur kimia seperti itu, kita tidak bisa mengandalkan pencarian menggunakan keyword ya Bapak Ibu. Ya, dan disinilah ketika kita menggunakan SciFinder, kita masukkan, jadi sebagaimana saya jelaskan di awal tadi, SciFinder memiliki fitur pencarian literatur berdasarkan struktur. Jadi ketika kita masukkan, kita gambar struktur kimia yang kita dapatkan ke platform itu, kalau misal di sana tidak ada, berarti baru. Kalau misal di sana ada, berarti tidak baru. Jadi itu sebagai gambaran pertama, Bapak Ibu, apa yang menjadi keunggulan dari platform SciFinder ini. Jadi pertama, bisa untuk mencari tahu kebaruan dari senyawa yang sedang kita teliti. Kemudian yang terkait dengan proses pengembangan obat, sebagaimana saya jelaskan tadi, yaitu modifikasi struktur kimia untuk meningkatkan keampuhan dari obat tersebut dan menurunkan efek sampingnya, itu kita perlu melakukan modifikasi struktur kimia. Kita perlu melakukan reaksi kimia terhadap senyawa tersebut. Itu juga menjadi pertanyaan, reaksi apa yang mau kita lakukan? Bagaimana mencarinya di literatur? Itu juga menjadi pertanyaan. Jadi menggunakan platform SciFinder ini, kita bisa eh, menempatkan. Jadi salah satu fitur yang ditawarkan oleh SciFinder ini adalah pertama kita bisa melakukan pencarian berdasarkan struktur. Kemudian yang selanjutnya struktur tadi bisa kita spesifikkan lagi. Misal bisa kita spesifikkan lagi perannya sebagai bahan baku starting material. Jadi kita bisa tahu dari sana senyawa yang sedang kita lihat ini tuh pernah diapakan aja sih sama orang-orang, sama periset terkait di literatur seperti itu, Bapak Ibu. Dan ini menjadi penting karena ketika kita melakukan pengembangan obat, kita perlu tahu konteks ya, atau posisi dari penelitian kita itu ada di mana di dunia. Jadi secara umum seperti itu. Dan selain terkait dengan senyawa kimia yang secara spesifik disebut senyawa small molecule, Bapak Ibu, di SciFinder ini juga menyediakan fitur yang serupa, tapi terkait dengan kalau tidak salah itu tentang ini ya, tentang DNA dan semacamnya ya. Ini kebetulan di luar kepakaran saya, jadi saya belum pernah menggunakan fitur tersebut. Cuman kalau ketika saya baca di guideline-nya itu ada, dan di tampilan interface-nya itu ada, mungkin nanti Bapak Ibu di bidang riset terkait bisa mencobanya. Dan saya juga sebetulnya penasaran ini, nanti ingin menyimak dari pemateri yang kedua setelah saya, karena beliau ini memang expert dari SciFinder-nya sendiri. Kalau saya hanya menjelaskan dari sudut pandang saya sebagai kimiawan organik sintesis. Kemudian dari proses dari deskripsi dari contoh kasus tadi, Bapak Ibu, bisa dibayangkan ya, misal kita tidak punya akses ke database untuk mengetahui seberapa baru senyawa yang kita sedang teliti dan modifikasi struktur seperti apa yang sudah pernah dilakukan di literatur, kita akan sangat sulit untuk mengejar kebaruan, juga untuk mengetahui posisi riset kita ada di mana. Jadi itu satu poin penting yang perlu digarisbawahi. Kemudian bidang riset apalagi yang bisa memanfaatkan fitur-fitur yang ada di SciFinder ini? Sepemahaman saya, Bapak Ibu, sebagai seorang kimia, itu riset-riset yang terkait dengan senyawa kimia, struktur bahan kimia itu seharusnya bisa memanfaatkan platform ini. Contohnya, sebagai contoh lagi itu bidang-bidang riset yang terkait dengan pengembangan energi. Mungkin untuk energi ini saya ambil contoh kasus saja tentang ini ya Bapak Ibu ya, di negara Indonesia kita ini kan banyak menerima paparan sinar matahari ya, sehingga pengembangan solar cell di negara kita ini cukup menjanjikan. Kemudian secara lebih spesifik lagi, solar cell itu mungkin Bapak Ibu yang di tema riset ini lebih paham ya. Untuk yang generasi pertama itu kalau tidak salah ya kan yang berbasis silikon ya. Namun karena bahan ini perlu dibuat pada temperatur tinggi dan semacamnya, sehingga perlu dicarikan alternatif lain ya. Kalau tidak salah muncul istilah solar cell generasi kedua yang berbasis senyawa-senyawa organik. Dan juga ada yang berbasis tentang ini perovskite. Kalau perovskite saya kurang begitu paham. Nah, tentang solar cell yang berbasis senyawa organik ini sangat terkait dengan struktur kimia ya Bapak Ibu ya. Jadi dari yang pernah saya baca ini, istilah organic solar cell ini dibuat dengan lapisan aktifnya itu berbahan dari senyawa organik yang disintesis. Nah, dari situ kita juga bisa memanfaatkan atau kita juga memerlukan step-step berupa pencarian senyawa apa ya yang sudah banyak dipakai sebagai active layer untuk organic solar cell. Sudah sampai mana, efisiensinya sudah seberapa, dan modifikasi yang dilakukan sudah seberapa jauh. Dan ini sebagai informasi tambahan lagi Bapak Ibu, sepengetahuan saya senyawa-senyawa kimia itu ketika di literatur cukup jarang ditampilkan dengan nama aslinya ya kan, ada penamaan senyawa kimia itu berdasarkan ini ya kesepakatan bersama yang dinamakan ini menurut aturan IUPAC itu ya. Jadi kalau kita menggunakan nama itu namanya sangat panjang. Biasanya dia diberi nama inisial seperti NS22 apa seperti itu ya Bapak Ibu. Jadi untuk mencari sekali lagi untuk mencari literatur terkait senyawa kimia kita perlu perlu platform yang mana kita bisa memasukkan inputnya itu berupa struktur senyawa kimia. Tidak bisa hanya dengan kata-kata. Mungkin ada, kita dapat bisa, tapi tidak secara eh, jaringnya itu kurang kurang lebar ya. Mungkin ada referensi-referensi yang tidak kita dapatkan dan itu pernah saya alami sebelumnya ketika saya belum menggunakan SciFinder itu saya juga bertanya-tanya ketika saya dapat senyawa seperti ini dari mana bisa tahu ini ada di literatur atau tidak. Gitu. Jadi ini sebetulnya sangat memudahkan. Kemudian aplikasi lain Bapak Ibu yang terkait yang bisa eh, yang bisa memanfaatkan platform SciFinder ini adalah dari kacamata saya ini di bidang material ya, material maju. Ini dari beberapa tema yang saya baca ini eh, terkhusus tentang saya ambil contoh saja tentang nanomaterial. Nanomaterial yang berbasis senyawa organik. Sekarang ada yang sedang naik daun ya, dia berbasis peptida. Itu peptida juga sekali lagi adalah senyawa kimia. Dan kalau kita mencari di literatur menggunakan kata-kata, kita mungkin dapat, tapi tidak semuanya. Jadi, dan itu menurut saya jadi sangat tidak efektif ya Pak Ibu. Jadi, seperti itu gambaran yang bisa saya sampaikan. Dan sebagai informasi juga sepengalaman saya ketika berada di luar negeri itu ini ada ini ya, mohon maaf. Sepengalaman saya di universitas-universitas yang memiliki kegiatan penelitian terkait dengan kimia, mereka pasti berlangganan SciFinder ini karena SciFinder ini bisa dibilang adalah sumber informasi yang utama yang digunakan oleh periset-periset tersebut untuk mencari literatur. Jadi langsung cari di situ ada atau tidak, kalau tidak ada berarti baru atau mau cari metode apa di sana ada. Jadi mudah-mudahan bisa memberikan cukup bayangan mengenai teknis detail penggunaan SciFinder seperti apa, mungkin akan disampaikan oleh pembicara berikutnya. Kemudian untuk informasi berikutnya yang mau saya sampaikan itu adalah mengenai pembuatan akun ya Bapak Ibu. Pembuatan akun di platform SciFinder ini nanti bisa saya share ini tutorial singkatnya. Jadi kita nanti mendaftar begitu, create account, lalu kita memasukkan email BRIN kita dan kita tinggal mengikuti langkah-langkah berikutnya. Jadi sebetulnya cukup simpel, nanti bisa diikuti bersama. Ini coba saya izin sampaikan di kolom komentar sebentar.
Share screen, Pak Yudi.
Oh boleh.
Share screen saja.
Saya screen saja ya. Oke. Ini, mohon izin untuk share screen. Oh ini kebetulan saya bisa dibantu diberikan akses sebagai co-host mungkin untuk bisa share screen. Bapak Ibu. Mohon izin untuk share screen tentang tutorial pembuatan akun. Berikut mudah-mudahan sudah tampil di layar Bapak Ibu sekalian. Pertama, kita buka link berikut ya, bagaimana ditampilkan di nomor 1. Kita buka link ini. Kemudian nanti kita pilih, nanti tampilannya akan seperti ini ya Bapak Ibu, tampilan interface awalnya. Kemudian Bapak Ibu nanti bisa pilih yang create account ini. Create account. Kemudian setelah klik create account, nanti akan muncul pilihan, ada 2 pilihan. Nanti kita pilih yang kiri ya Bapak Ibu, yang commercial or enterprise. Setelah kita pilih yang kiri, nanti akan diminta untuk memasukkan ini email. Jadi kita masukkan email BRIN kita masing-masing, kemudian kita klik continue. Pada tahap selanjutnya, nanti kita akan diantarkan ke page berikutnya, yaitu berupa kode yang harus kita masukkan ya, kemudian kita klik continue. Jadi setelah kita tadi klik continue pada step nomor 4, kita akan menerima kode di email BRIN kita, mungkin kurang lebih antara 1-5 menit kodenya masuk. Kemudian setelah kita masukkan ke sini, kita klik continue. Nanti akan muncul seperti ini. Sebentar. Tadi masukkan kode, lalu kita klik continue dan nanti kita akan membuat akun. Di sini bisa kita pilih, ini prefix ini adalah kalau tidak salah Mr. atau Mrs. Ini nama. Kemudian untuk institution name-nya saya waktu itu isi National Research and Innovation, di sini sudah muncul. Kemudian isian yang berikutnya itu ada job role. Kalau saya pilih scientist ya, karena sebagai periset. Di sini ada macam-macam, nanti bisa dibaca sendiri. Kemudian isian berikutnya ini tentang area riset. Ini nanti bisa disesuaikan dengan area riset Bapak Ibu sekalian. Kalau di kasus saya pilih chemistry. Kemudian berikutnya yang bagian bawah ini ya, yang kontak phone dan ICS member ini opsional. Jadi cuma saya tidak isi waktu itu. Jadi kemudian saya klik request access. Setelah itu nanti kita akan menerima email dan kita tunggu nanti. Ini tampilan emailnya sepertinya ada di bawahnya ya. Nah, nanti kita akan terima email, ada yang bisa kita klik ya, create your username and password. Ini bisa kita klik langsung. Kemudian nanti kita akan membuat username kita sendiri dan password. Ini ada petunjuknya. Petunjuk pengisian. Jadi username-nya berapa karakter, harus ada apanya, itu nanti bisa dibaca sendiri, ditemui, dan passwordnya juga. Kemudian kita klik di sini juga ada security question ya. Nanti Bapak Ibu bisa pilih sendiri mau pertanyaannya apa. Mungkin ini nanti akan dipakai kalau kita lupa password mungkin ya. Juga beserta jawabannya kita tuliskan di sini. Kemudian kita klik login. Setelah itu setelah itu sudah selesai, kita masukkan username yang tadi kita buat, kemudian kita klik next. Dan kita masukkan password yang tadi kita sudah set. Kemudian kita sudah punya akses untuk memanfaatkan fitur-fitur yang ada di SciFinder ini. Mohon maaf tadi saya tidak menjelaskan detail tentang fitur-fitur yang ada di dalam karena akan dijelaskan oleh pemapar berikutnya. Ini nanti akan saya share di kolom komentar untuk bila Bapak Ibu masih ada pertanyaan. Mungkin demikian dari saya, Pak Indra. Kalau misalnya ada pertanyaan, nanti bisa disambung di sesi tanya jawab nanti. Terima kasih banyak. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Mas Yudi atas sharing sessionnya. Selanjutnya, marilah kita mendengarkan penggunaan SciFinder untuk preset BRIN yang akan dibawakan oleh perwakilan dari CIS SciFinder, dan kegiatan ini akan dimoderatori oleh Bapak Wendy Purnama selaku Academic Sales Manager at iGroup Asia Pacific Ltd. Kepada Bapak Wendy, kami persilakan.
Iya, baik. Terima kasih banyak atas waktu yang diberikan pada siang hari ini. Apakah suara saya terdengar?
Terdengar, Pak.
Dengar ya, baik.
Ya.
Baik, pada hari ini saya akan memberikan rekan kami dari CIS SciFinder, Pamela Un and Adran Ling from CIS will introduce about the CIS SciFinder and how to use and utilize the SciFinder. Okay, without further ado, Ms. Pamela, maybe you can start to explain about the CIS SciFinder.
Yeah, okay. Thank you, Andy. Very good afternoon, everyone. I'm Pamela from CAS, and I'm based in Singapore, the same office as Adrian. So I'm going to bring you through CAS SciFinder Discovery Platform. So please allow me to share my screen first. Okay. So you should be able to see my slides. Okay, so I'll start with a brief introduction on CAS. So basically, CAS, we are one of the divisions of the American Chemical Society. And what we do at CAS is curate and organize scientific literature. So it includes journal information as well as patent information. Over here on the left-hand side of my slides, you can see some different keywords that represents different research areas. So in terms of our content, we have obviously chemistry, which we are really good at, but we also have information from life sciences, from material sciences, and a lot more. And you can see that these days, different research areas, they are very interconnected. So it is important that you use a database that's able to present these connections and relationships to you. Okay. And one of the key advantage of using a database like SciFinder is that the content that we put into our database, the CAS scientists actually reads through the full text journal articles or patent documents. And in this process, we index the important information disclosed or described in this literature. So it could be a novel concept, it could be a new compound, it could be formulations or chemical reactions. So all this data is actually extracted out from the full text, and we add it into SciFinder so that you can actually search for a substance. You can search for keywords or you can search for formulations. Okay. So just an example of how much content is found in just a single document. So this is a patent example. So just from this one document, the CAS scientists actually extracted out 7 novel concepts, 138 substances that were involved in the invention of this patent, over 4,000 reactions. So this vast amount of data is made easily discoverable via our platform. Okay. So this is a kind of a summary of different types of information that you'll be able to access or search for via the CAS SciFinder Discovery platform. So I've mentioned references, pattern, as well as journal literature. We have substances, over 250 million substances, and it ranges from your small molecule to polymers to inorganic compounds, even including protein and DNA sequences. You have reaction information, and we also have Marcouche structures. You can also find regulatory information, and we also provide bioactivity data, analytical protocols, and formulations, chemical suppliers, and a lot more. Okay, so in my introduction of the platform later on, I would try to kind of cover most of what what you're seeing here. I won't be able to cover everything, but I'll try to show you as much as I can, okay, with the time I have. Okay, so before you could use the platform, as Dr. Yudi has mentioned earlier on, you do need to first register for an account using your BRIN email address. So once you've done that, okay, and you log on to SciFinder End, this is the homepage that you see. So I've mentioned that as part of the SciFinder Discovery platform, you have analytical methods as well as formulations. So if we go to the three black dots found on the top left-hand corner, okay, this is what we call the app switcher. Click on it, and you will find that under SciFinder Discovery platform, you have SciFinder N, which is the platform that we're looking at right now. And you have the links to access two other databases, okay? So I'm just going to click on it to show you how it looks like. CAS Analytical Methods. So this will be a platform for you to search for analytical protocols, analytical procedures, all right? And then the other one would be Formulas, which is a database that's specific for formulation information. Okay, so from SciFinder End, you can access the other databases. And if it prompts you for your username and password, just use the same username and password that you have used to log on SciFinder End to access the other two databases. Okay. All right. So now that you know, okay, this little hidden button over here, all right, for you to access the two other databases as part of the SciFinder Discovery platform. So let's now take a closer look at SciFinder End. So on the home page, you can already see that you can do different types of searches, okay, including retrosynthetic analysis or a blast search for sequences. So we're going to look at reference searching first. Okay, and you can type in your main search topic into the box here. If you click on the drop-down list, it will allow you to do reference search via other starting points, like publication name, author name, etc. And you have always the option to combine multiple search terms into a single search. So let's look at a simple keyword search example. So I'm going to type in my topic of interest, which is the name of a plant. And you can see as I'm typing out, I do get the word suggestions. If I find something that is relevant, I can click to add it to my query. Okay, and this will be my topic. So I'm going to go ahead and run the search. Okay, so this is the results. And as we scroll through the results, we do see blue hit highlighting that matches what we have looked for. Okay, in some cases, you may notice that For example, this reference number 4, it seems to be only matching partially of what I'm looking for, right? If you see cases like this, one of the things you can do is to change the sort option. So right now it's sorting it by publication date. So we can swap it to sorting it by relevance. So when you do this, you will be able to find the most relevant documents now brought up to the top of the answer set. Okay, so now if I scroll through, you would see that the documents now are a better match to what we're looking for. Okay. The other thing you may notice is that for some of the references, there is this analytical methods link. So when you see this link attached to a reference, it's telling us that the analytical procedures reported in this paper can be viewed via our analytical methods platform. Okay, so if I click on this, actually, let me open this in the new tab. It brings us into what we call the detailed view of the reference. And right here, we have the analytical methods section, where in this paper, CAS has extracted out 4 procedures that you can view via our analytical methods platform. So on our results page, on the left-hand side, there is actually a filter that you can use to find out which papers have a method that you can access via analytical methods. So if I scroll down to the bottom, you can see here CAS solutions. All right, and it tells us that there are 22 references with analytical methods. So I can check this. Okay, so now with the 22 results, we have, you can see that they all each have the analytical methods link. Okay, so let's open up maybe the second one. Okay, I'd like to open this in a new tab. Okay, so quite a few methods that are available from this reference. Before we go into the analytical methods, I want to talk about the other sections that you're seeing here. So this is the detailed view of this reference, where you can find the information that CAS scientists have indexed from the full text. So remember in my introduction, I talked about it. So we have concepts which would represent the standardized terminology used by CAS to describe the research focus of this paper. And you can see the blue hit highlighting occurring here. So besides title and abstract, SciFinder actually searches concepts as well. So you actually get to search the important keywords that that describes what's important in the full text. Okay, then we have here substances. So these would be key compounds, compounds that are important in the research of this paper. So such as this first substance here, you have the structure, you have the name, we even describe the role. So the role tells us how this compound is actually being utilized in this paper. Okay, so it says analyte. So likely this is one of the compounds that have been analyzed in this publication. With all the structure images that you see here, you can always click on the image. Okay, it pulls out a pop-up window, and you can run a further search on this compound, whether it's getting additional literature, chemical supplier information, or finding out how to synthesize this compound. All right. Let me go to page 2. We'll have more substances there. Okay, so the same thing. We have here the structure with the analyte role. I'm going to click on this. And for this particular compound, you can even get bioactivity data. All right, so I'm going to click on this and show you how it looks like. Okay, so I am moving into substance information from reference. All right, so this is the compound that we were looking at over here. And we are interested in bioactivity data. So for this compound, there are two types of bioactivity data. We have the SAR data as well as pharmacokinetics data. So this is part of our CAS Life Sciences information. So let's give it some time to load. Maybe while it's loading, I'll go back to here. And if you find any interesting structures, you like to do a structure search on it, you can also click on Edit Structure. And this actually transfers the structure image into the CAS drawer. And from here, you can run structure searches, or you can make further modifications on this chemical structure. Okay, and we'll talk about structure searching later on. Okay, let's see if the data is loaded. All right, we have here pharmacokinetics data. All right, and on top over here, we have different bioactivity data for this compound. We could click on the filter button and look at different biological targets. So we'll be able to take a look at this again when we do substance searching. So just wanted to show you how we can easily move from a reference document into substance information. Okay. The other thing I would like to mention, you'll see here we have Medline medical subject headings. So in SciFinder End, other than the references that are curated by CAS, you get to search a second reference database, Medline. So I believe for the people who are working in the life sciences, biomedical areas, you would probably be quite familiar with Medline. It's part of the PubMed website, right? So you can search the Medline references as well as the CAS references at the same time using SciFinder End platform. So it's very convenient. Okay, now moving down to analytical methods. Okay, so we can see here there are quite a few methodologies that have been indexed in our analytical methods platform. When you look at the title, it looks like they are identical procedures, but actually it's not. Okay, so whenever you're trying to decide if the analytical method is relevant, I would suggest to go ahead and click on the links shown here to review the method before deciding if it's relevant. Okay, so I'm going to click here. All right, so when I click on the link, it actually brings me to the analytical methods platform. Okay, like I say, if it's the first time you are making the transition over to the platform, it may prompt you for username and password. Use your SciFinder and username and password. So this is the title of the method. But actually, if you look at the information here, it doesn't only analyze this compound. There are actually quite a few analytes involved in this analysis. It doesn't only talk about solvent extraction. In terms of the technique, it involves solvent extraction as well as microplate spectroscopy. So that's why I say if you're trying to decide if the methodology is relevant, don't just make the judgment based on the title. It's always best to click on the method and then check out the full details of the analysis. Okay, now as I scroll down, you will soon be able to see the details of the analysis. Okay, so it tells us what equipment was used, and then it goes on to say how they prepared a sample, how was the solvent extraction done, and then finally, this is actually an essay that is to determine the antioxidant activity of the extracts. Okay, so we even have the validation data reported, or the bioactivity data reported from this analysis. So this information here, you typically would have to go to the full text to get the details. But right now, with analytical methods, you can actually have access to this information, even if you don't subscribe to the full text of this particular article. Okay. So, the same thing, if you want to see the details of the other methodology, just click on the links accordingly. Now, eventually, if you do decide that you want to go to the full text of this paper, SciFinder, it's not full text database, so we don't provide you with the full text, but we have the links that you can use to access full text. So I can click on DOI here. It would typically bring you to the publisher's website, okay? And then depending whether is it open access or if your organization subscribes to it, in this case, I can see it's open access. So you will actually be able to look at the full text in this instance, okay? So if you need to go to the full text, you can use one of the links provided here to access, to potentially access the full text. We also have a similar references function here where it would suggest other documents that are similar to the one that we're looking at. You can scroll through the panel, okay, to see if there's anything interesting, or you can click on Get Similar References. So it will suggest up to 500 similar references. It can be less than 500, but a maximum of 500. Okay, and then you can view through all the 500 documents over a new tab as well. Okay, so now I'm going to hit back into our search results. So we've looked at analytical methods. Okay, so let me clear this filter. So the other useful filter that you can use if you're interested in bioactivity data is the bioactivity data filter here. So there are two documents with bioactivity data. I will select it so we know which ones it is. Okay. So for these two documents, if you want to look at bioactivity data, you'll have to, again, go into the detailed view of the reference. So I'm going to open this in a new tab. Okay, there's no like extra link like analytical method. So just click on the title, go into the detailed view. So in the detailed view, you get to see concepts, right? Medline, medical subject headings, substances, just like before. We can expand it. You can see here, these compounds, they have now pharmacological activity role, right? Earlier on, we saw analyte role. So this has pharmacological activity. So these are likely the ones where you're going to find bioactivity data on. Now, I've scrolled down further. Here we have the CAS Life Sciences data. So I can expand this. Okay, so we have here the table of bioactivity data. These are the ligands. Okay, with the bioactivity data, you can hover over the CAS registry numbers to see the structures. And again, if you're interested to find more information on any of the compounds, you can click on the CAS registry number. We get the pop-up window, and then from here you can get further information on any compound that you see in this section. Okay, so CAS Life Sciences Data. Let us go back again to the results. Okay, so we were using the bioactivity data filter. Okay, then the other filter that I want to show you would be formulations. Okay, so earlier on with the CAS solutions filter, we can see that there are 5 references that have formulation information. There's another filter that would tell us which references have formulation, which is the formulation purpose filter. So with this, we can select formulations with a certain objective that aligns with what we're looking for. So I'm going to select this for here. Okay, we're looking for formulations for facial products. Okay, so this for two papers here, but then there could be they match kind of these 4 formulation purposes. The same thing, if you want to look at the details of the formulation, you have to go into the detailed view of the document. In this case, we have a patent document, and you notice there is the patent pack icon. So for patent documents, for most of the patent offices, the major ones at least, you would typically find the patent pack function available, which will then allow you to actually download or access the full text PDF of the patent directly from SciFinder end. Okay, so I could click on this or any of this to look at the full text patents. I could also go into the reference detailed view again. Okay, which is this. Just let me double check. Yep. All right, so this is the reference detailed view for the patent, and we have a patent from L'Oreal. And again, we have the various links for you to access the PDF of the patent. And right at the bottom, we have here formulations. Okay, so this patent reported quite a few formulations that CAS have indexed in our formulas platform. We can open it up to take a look at, what are the ingredients used, what are the amounts. But this is just a preview of this formulation. To look at the full details, because you can see here there are actually additional components reported that we don't see here. So we can click on CAS formulas detail, and then this brings us into the formulas platform. Okay, so right now I'm in formulas, and I can expand all groups to see the full ingredient list of this formulation, and it even reports the process of preparing this formulation. Okay, so all this data is available in the full text. Of course, in formulas, you could click on this to download the full text pattern, or if we do it from SciFinder end, let me show you the viewer version. Okay, so this is just the regular PDF, but the viewer version contains key substances. Okay, on the left-hand side panel, as you can see, and it's marked up with page numbers. So if you're interested in this compound, for example, we can go into click on page 8, and then it shows us where this particular compound is mentioned. Okay, and the markers actually work both ways. So if I'm looking at one of the formulation for a care cream, I want to know more about this substance, I could click on the marker and it would tell me, okay, this substance here is actually this compound. And if I want to do get more information on any of these ingredients, I could then click on the structure image, and this again brings up the pop-up window for me to get other information around these compounds. Okay, so this is using the viewer version of the patent pack. If you click on this and this, it will just kind of give you the PDF, just a plain PDF without the substance information on the left-hand side. All right. Okay, so from here, we've seen quite a bit of different data that you can access from just one search. You can access analytical protocols, you can access formulations, and you can also access bioactivity data. And we've also seen the patent pack function for us to access full text patents. So now I'm going to take a look at another topic. So you can run a brand new search using the search bar on top. So let me clear this. And I'll type in another query. This time, I'm going to do a more specific search. I'm actually going to incorporate Boolean operators and other symbols to make the search more specific. Okay, so I'm interested in CO2 hydrogenation. So I'm putting it in double quotation because I want a CO2 hydrogenation to be searched exactly as a phrase like this. I have a synonym for it. I also wanted to search CO2 reduction. So in this case, I put in the CO2 reduction with double quotation again, and I use the Boolean operator or, because I'm happy with either phrase, so either or. And I have another concept or topic I'm interested, which is to look for photochemical or photothermal type of reactions. So I can search photo, and to keep my options open, I put in the wildcard symbol, asterisk. So like I say, it can search for photo, chemical, photothermal, or photocatalysis. Okay, and I'm putting in another keyword, catalysis, and I'm going to use let's put in the Y. Okay, and I put in the asterisk again because maybe I want to search catalytic, catalyze, catalyst, etc. Okay, so this is a combination of the and or operators with double quotation and the asterisk. So I'll go ahead and do the search now. So these are the different operators that you can incorporate into your search query. So you have more control over the results that you see, right? Whether, how specific does it need to be, or in some cases, you want it to go a little bit broader, you can use the asterisk. Okay, so this is the results. And in this case, I would actually, so let's just give it a couple more seconds. It's to load everything up. Okay, so this is now fully loaded, and there is a load more results button. So in the first example, I didn't click on this because my search query was not specific. Okay, and I was already getting like partial matches. But in this case, because I have indicated clearly which words are compulsory, which words, I'm allowing some variation, I would go ahead and load the entire result set. Okay, so this will be the full result set. from 9,000 plus to 10,000 plus results. Okay, so in this case, even if I change my sort option to publication date, right, I no longer use the relevance ranking, but because my search query is specific enough, I would still find my required terms appearing in the title, in the abstract, or in the concepts and substances section. Okay, so let's take a look at another filter option that is really useful, and that would be the concept filter. So you've seen CAS concepts already. So this filter here allows you to have an overview of all the important concepts for the references in your results. Okay, so you can see we didn't search X-ray photoelectron spectra, but it's actually the third most commonly occurring concept. So this would give you an idea or a hint that for this area of research, this is one of the techniques that people could use to maybe characterize the catalyst that's being researched on. Okay, but regardless, this is an important technique. Things like surface area is important, nanoparticles, absorption, right? These are all related topics to what we have just searched for that are important. That's why they occur frequently in our result set. Okay, so using this top count list, like I say, you can see what are the other related topics that are important. And if you find something interesting, for example, metal organic frameworks, we can select this concept. So now it will help us focus our results to papers that, other than looking at CO2 hydrogenation and reduction, it also has a focus on metal organic frameworks. All right, now let's take a look at more details of this particular reference. So I'm going to click on the title, open it in the new tab. Okay, so the same thing, we'll see concepts, substances. All right, and you notice in this case, the role is a completely different role from what we saw earlier on, right? Earlier on, we saw analyte, we saw pharmacological activity, but for this area of research, we see catalyst use role. So by looking at the role, you know which of the substances here are used as the catalyst or part of the catalytic system being investigated in this paper. Okay, and then if the structure again is too small, click on it so that you can see expanded version of it. You can continue to zoom in if you like, and you can even download structure images in different formats over here. Okay, and then the other role that you can see, synthetic preparation. If you see this role, then it would indicate that in the full text, they likely reported preparation process for each of these substances that we see here. Okay, and of course, we selected metal organic frameworks as the concept. So we have here metal organic frameworks. So when you use the concept filter, it is looking for relevant references based on the CAS concepts. Okay. Now going back again to our result set. So let me open up concept filter. Let me uncheck this first and we'll open this up. You could also run searches within the concept filter to find a concept of interest. Okay, so I can click on the search tab, and maybe my interest is to see if there are any papers that use zeolites, right, as the catalyst. So I can search for zeolite, and it will pull back all concepts that are relevant or related to zeolites. So you can see we actually have very specific terminology used for different types of zeolites, and we also, of course, have just the generic zeolite concept. So with the concept filter, it can help you to really get a lot of precision in terms of identifying relevant documents, okay? And if you wish, you could always select more than one concept. Okay, so this is the concept filter. And then one last filter that is very useful is the search within results. So this is where you put in your own keywords. Because sometimes your keyword may not be a CAS concept, so you can't actually use this filter option. Or it could be that, you know, you want your filter to also take into consideration title and abstracts, because remember, concept filters only use the concept information. So I could go in and say, well, I want to find papers that look at the sol gel process, okay, of maybe preparing the catalyst. So I could search sol gel. Again, I'm putting in the double quotation for more precision. Do a search. So it will now look for the term or the phrase sol gel, and you can see now it's highlighted in the abstract or the title. It could also be found in other parts of the reference detail. Okay, so this is the other useful filter to use. Now, once you have run your search, you're happy with the results, you can actually save your search strategy so that the next time you don't have to reapply all the filters. So to do that, just click on this button over here, click on save, give it a name. You can choose to just save the search strategy or save the strategy together with the results. You can also set up your alerts over here so that when anything new enters the database, you will be informed. Okay. The other thing you could do is if you find relevant references, you could save them into project folders. So if you click on the 3 dots here, you can see here I have quite a few project folders already set up. You can drop them in existing folders, or you could always create a new project folder and organize your saved answers really nicely, especially if you're working on multiple projects. So whatever that you have saved, right, whether it's projects or your search strategies, you can access them using this little icon over here called Saved and History. So I'll open this up to show you. Okay, so I have here search strategies that I have saved previously. And I can access my projects as well. Okay, my project folders. I can click on the folder name and you'll be able to see the references that you have put in previously. Okay, and you could add references as well as substances into the same project folder. If you have set up alerts, right, if anything new enters the database, you will be notified over here. So you can see here I got a little number 2 here, which is telling me that I have some new alerts that I have not looked at. So when I click on alerts, okay, I'll be able to see which are the new additions, okay, for the alerts that I have set up. And then finally, we have the history tab, which it automatically saves the searches that you have run, the initial searches. So these 2 searches I've run so far automatically saved for me. And you can even use the calendar here to access the search history from months and even years ago. Okay, so it's this calendar. Okay, now let's move on to substance searching. So hopefully, you know, this is a clear, right, for reference searching. So I'm going back to the homepage and let me close some of this. All right, so moving into substance searching. Substance searching, you can do it by name, you can do it by structure or even molecular formula, and we have, NMR chemical shifts, or you can find compounds by various different physical chemical properties. So I'll do a simple search, substance name search first. Okay. So again, as I'm typing, I see the word suggestions. If I find my compound, I can select it, run the search. Okay, so this is the substance that I'm interested in. You can link out easily to references, reaction, and even chemical supplier information. You can also click on the CAS registry number, and here it will bring you into the detailed view of the substance, where you could find things like the bioactivity data we saw earlier on, as well as spectra and other properties. Okay, this is the GHS hazard symbols. So this tells you the associated hazards related to this compound. I can click on this, and then it tells me what are the hazards. So this is quite useful, right? So before you run any experiments, you can kind of find out what are the potential hazards that you'll be exposed to. This has structure activity, pharmacokinetics, as well as toxicity data for narogenin, okay? So this will be all the bioactivity data from different papers that are available for this compound. All right, so like I mentioned earlier, for especially when you have pages and pages of data, use the filters at the top to help you locate the type of data that you are interested in. Okay, let's see what else we have here. Okay, we also have experimental spectra data for narogenin. All right, sometimes the spectra data is not available directly in SciFinder N, but we will tell you which sources you can go to get that information. If it's available, you'll see that, it's a clickable hyperlink, okay? Similarly, for experimental properties, we may see things like see full text, okay? And then you have to go to the individual sources to get that information. Okay, so this has loaded up the bioactivity data. So this is quite a few pages of data. So you definitely want to consider maybe filtering by specific targets. Okay, we can even expand it and look at all the different biological targets that we have bioactivity data for. And if you click on view assay detail, you have a little bit more information on what the assay was. Was it a in vitro, in vivo, things like that, okay? All right, so this is searching substances by name and the type of data you can get for a substance. And you can always link back to literature, right? So we have this icon here, which will pull back references for this compound. So when I do this, it brings me back to the reference answer sets that we were looking at earlier on. Okay, and in this instance, it's actually quite useful to use the substance row filter. So I didn't talk about this earlier on, but now we have all these results that are related to this compound, but they cover many research areas, right? So not everything here is going to be relevant. So the substance row filter is one quick way to narrow down the references to the application of the compound that you're interested in. So if you're interested in bioactivity data, you could select pharmacological activity. If you're interested in analysis, you can select analyte. If you're interested in natural product type of research, you can select natural product occurrence, okay? And then it will filter it accordingly. So let me just kind of show you. Okay, so let me just open up one example. So we will see our narogenin appearing in substances, and then assigned natural product occurrence role, as well as other roles that are relevant in this paper. Okay. Now, if we want to do a structure search, right, using the same compound narogenin, we can, like I mentioned earlier, edit structure, add it into the drawing editor, and I'll talk about the draw editor in a moment, but let me go ahead and run the substance search first. So the substance search is a lot more powerful because it allows you to find not only the compound that you're looking for, but also similar and related compounds, right? It could be analogs, it could be mixtures or salt forms. So you can see here, with the structure of narogenin, we actually have 3 types of answer sets, right? The estron would be substances that match the same structure that we have in the structure query. The reason why we have more than one answer is because it also captures stereoisomers, although in this case, we have only selected absolute stereo match and absolute stereo mirror image, right? So you can even choose to just focus on the absolute stereo match. You may see deuterated compounds like this. You may see ion versions, free radical versions, salt forms, okay, hydrates, mixtures, right? So all these would come under estron, and they could be interesting because it all still involves narogenin, okay? Now, in the second answer set is where things get a little bit more interesting, the substructure. This is where you would see some modifications to the structure of narogenin. So it will still include narogenin, I mean the answers from the estron. So we can actually filter this down a little bit. So let me first focus on single component answers just to keep things simple. I could remove the the isotopes. So we have an isotope filter, not containing isotopes. Okay, let's see. All right, so this compound over here, number 15, is the first example where we start to see modifications on the structure of narogenin. So I don't know if you notice, this compound has an extra methyl group on the oxygen, right? So whatever that's highlighted in blue matches the original structure we put in the query. This is the modification. So as I scroll down, you kind of see more of these compounds. And the modifications can come on any part of the molecule, right? It could be on the aromatic ring. Okay, this is like additional OH. This is on a different methyl group on a different oxygen. Okay, so this would be close analogs of narogenin that could be interesting. And with this large answer set, we do have many filter options that you can use. I've already shown you some of them. Some additional ones you can consider filtering by bioactivity data. So which of these compounds actually have bioactivity data? This filter will tell you which ones. Commercial availability. You could also maybe refine by number of aromatic rings. Right now we have 2. Maybe you just want to go up to 1 additional aromatic ring maximum. So I can select 3 aromatic rings. And then we start seeing compounds with the 3rd aromatic ring appearing. Okay, we also have functional group. So you can choose compounds that have certain functional group, and you could also select compounds based on their hazards. All right, so remember there's always the exclude function, right? You can choose which compounds to keep. You can also use the exclude to remove compounds that you're not interested in. Okay, so with a substance search, as you can see, you get a lot more hits and a lot more information and related compounds. Okay, now let's look at another way of searching for substances, which is molecular formula. So this would be quite useful for people working with inorganic compounds. So there is the regular way of searching for molecular by molecular formula. So for example, in this example, I'm looking for molybdenum with 2 selenium. So I can search it this way. All right, so I have here the substances that match 1 molybdenum with 2 selenium. Okay, and then we have this other type of compound, which is what we call the tabular inorganic substances. So usually for this type of substances, sometimes the ratio is unknown. Sometimes the ratio are in this sort of decimal places. So there isn't a proper molecular formula like in this case. Okay, so to search this type of compounds, right, it requires a different format for the molecular formula, but it can be quite interesting. So let me use another example. Okay, I'm going to remove this. I want to find substances that contains selenium, indium, and copper, right? But I do not have to restrict my search to a specific ratio. So to do this type of search, I can just type in indium, selenium, copper. Okay, just type out the elements that you're interested in, put in the period symbol after each element, and then you can run the search. So this will pull back everything in our database that contains selenium, indium, as well as copper. Okay, over 600 results. And they are either in an unknown ratio, in many various types of ratio. So this is a very broad search, but you can see what type of ratio compositions are kind of known in the database for these 3 elements. Okay, and the next thing you may ask is, well, these compounds look interesting, but what are they frequently reported for? So we can use the button at the top to pull back the literature for all the 600 results. Okay, so again, I'm going back into literature, and because we have the concept filter, so it's really good way for us to kind of learn about different research areas. So what I like to do when I'm kind of investigating on searching on new types of compounds, the first thing I'll do when I cross over into the literature, I would go into the concept filter and see what are the top occurring concepts, right? So I see solar cells, semiconductor materials, photoelectric devices. So I know right away, this type of materials, they are commonly used for these applications. Okay. All right, so this is substance searching. Now I'm going to move into reaction searching now. So go back to the home page. So for reaction searching, you can technically search by text, right? You can put in the name of your target molecule or the catalyst that you want to use or the reactant. But I think most of the time, for organic synthetic chemists, they would search by chemical structure. So let's take a look at how to use the drawing editor here. First of all, the drawing editor, the drawing space, you can maximize it if you need more space. It is quite similar to ChemDraw. So if you use ChemDraw, you should be quite familiar with this. And if your structures are already drawn in ChemDraw, you can consider importing it in. Okay, save your ChemDraw file as a mol file, or you can check if you have the add-in, the SciFinder add-in in your ChemDraw software. So I think it needs to be version 19 and above. If you have 19 and above, you can actually launch the SciFinder and search directly from your ChemDraw file. Okay. If not, you can always import it in, or you can convert your ChemDraw structures into a small string and then paste it in here to generate the structure. If not, you have all the tools you need to create the structures from scratch. So let's kind of go through the drawing editor. The commonly used elements are here. We have the full periodic table over here. Okay, so I'm going to convert the single bond to a double bond by clicking on the single bond 2 times. I could always switch to the double bond button if I want to. If I want to replace this with oxygen, right, because right now it's carbon, I can select oxygen here, or I could also use my keyboard and type O. All right, so this gives me oxygen as well, the same thing, I'm using O. And we do have the chain tool here. So if I need to draw kind of long chains, I could consider using this option. Okay, and then I'll continue building my structure. I type N on my keyboard for nitrogen. Okay, so this is my target molecule, and I'm just going to bring this over to the side and can add in the reaction arrow to give me the product. So I could run a search just like this, or I can consider putting in the structure, the reactant. Okay, the other useful feature in the CAS draw is, if this is something that you need to search frequently, you can consider saving it as a template in your own account. So this will be the button. Okay, give it a name. All right, click OK. And your saved templates can be accessed here. All right, so these are the standard default templates in CAS draw. Scroll all the way down. You have my templates. Okay, so these are structures that I have saved, and I can come in here to reuse them anytime, right? So I could just click, bring it to the drawing space, click again to add it. Okay, so just to repeat what I just mentioned, to create a template, click on this button, save it, give it a name, save it. To access your templates, go to this icon over here, scroll all the way down, okay, to access your own user templates. So I'm going to go ahead and run the search without any starting material. Let's run the search. Okay, so we have 2 types of answers. So the S-drawn will show us reactions that matches the end product that we have asked for. So you can see here, okay, including multi-step reactions. And in this case, for these 2 reactions, these 2 sets of reaction, if you want to know the procedure, you would have to go to the full text. Okay, and if it's from patent literature, then you can make use of the patent pack links to access the synthetic protocol. Now, if I move on to the second answer set, which is substructure. So substructure is the same definition as I've talked about earlier on. Okay, it would include our structure query as part of a bigger structure. So you can see the blue hit highlighting is where it matches our reaction query, right? But there are modifications at various parts of the molecule, okay, like you can see here. So sometimes this could still be useful. It doesn't match our molecule exactly, but it could be the modifications, if they're not at the reaction site, you could still consider the reaction conditions to for the synthesis of your own molecule. Okay, for reactions that have synthetic protocols, all right, we have this filter, so you could identify which reactions have synthetic protocol. If a protocol is available in SciFinder end, let me select it again. Looks like it didn't register. Okay, you will see this link here. All right, and then when you click on this link, you'll be able to view the details of the synthesis. Again, this is typically found in the full text. Okay, so now you have access to some of the synthetic protocols directly from SciFinder end. Even the characterization data. There are some useful filters such as reaction scale. So you can choose, if you're going to do a large scale reaction, maybe you want to consider kilogram. Many filters here for you to explore. Reaction notes is quite interesting as well because it allows you to pick maybe certain equipment that you want to use, or green chemistry, or biocatalyzed type of reactions. Okay, then the other tool function that's related to synthesis is our retrosynthesis. So especially if you're planning a multi-step synthesis for a compound, you can consider using our retrosynthetic analysis function. So this is my target molecule. Okay, I can select start retrosynthetic analysis, or I could go to the home page and start retrosynthesis from here as well. All right, so there are some plan options that you can set, the number of steps you want to see, the starting material cost, what is the most you want to pay for the cost of the starting materials. And then because our retrosynthesis also provides predicted pathways, you can choose whether you want the predictions to be based on common chemistry, or you want to consider the less common chemistry. So once you make your selection, just click on Create Retrosynthesis Plan. Then the algorithm will start to plan out the synthesis for you. So this can take up some time, especially for the predicted steps. So let me pull it out. I've already pre-run this, so I can't, you can see it's still searching. So I will open this so that you can see how it looks like. Okay, so you can see here, it plans out the synthesis for you. It estimates the yield and price for this route. And then you click on this icon here, you can look at alternatives for each step, as well as the evidence, right? So evidence would be if you decide to carry the reaction out in the lab, the synthesis out in the lab, here is the literature evidence on, or literature reference on how to do it. If you want to change any of the steps that you see here, let me turn on predicted steps so I have more alternatives. Okay, so you can go into the alternatives. If you don't like what is presented to you as the first option, you can go into the alternatives. Okay, you have experimental alternatives as well as predicted alternatives. You can pick something else. So I'll just select a different one from the list. So it will update the retrosynthesis plan with your new selection and recalculate the new yield and price. Because once you make a change here, the subsequent steps will change as well, right? So it updates accordingly. It recalculates the yield, new yield, and the new price as well. It's really expensive. So probably you don't want to go this pathway. You can go in and make another selection. And you can go in at every step to make changes until you're happy with the final synthesis. Okay, so this is the retrosynthesis function. And the very last thing, I'll just quickly introduce it to you, which is our biosequence search. So going back to our homepage, this will be a very quick introduction. So if you click on this, you can search, do a blast search on protein or nucleotide sequences. Okay, so you could either upload a FASTA file or text file to upload your sequence. I'm just going to type in a really short sequence just to demonstrate to you. R, G, W, G, N, G, C, G, L, F, G, K, G. Okay, this is my protein sequence. I want to find similar sequences, and I would like to search the NCBI sequence database as well. So once this is done, you could just click on search. All right, so it will be the search will be submitted to the history. You can see it's searching. So again, I pre-run this just to show you how the results look like. So this is basically how the BLAST sequence search results look like. You have your query sequence, what we typed in, and the matching sequence. You can either find the literature associated to this subject sequence, and in some cases, you'll be able to see the CAS registry number. Okay, so right now it's sorted by alignment identity, so these are all perfectly aligned, but of course you notice these sequences are much longer than my query sequence, so you can make use of the sequence length to maybe limit it to sequences that are not too long, say 20. Okay, and then you can also select the sequences based on the organisms that it's isolated from. Okay, hit apply. All right, so again, just trying to save time. So once I've hit the sequence length, applied the sequence length filter, you can see now much shorter sequences. Okay, and let's see. If you're interested in any of the references here, you can click on this button. If not, you can also make use of the reference button on top to pull back the literature for all the 79 results. Okay, so if you were to kind of ask me, so how is this different from running a blast search on the NCBI website? So I think for sequence searching, the main value is that you can very easily access the literature associated to it. And then once you're in the reference results, you can start doing various filtering. You can look at concepts. And then the other thing is the CAS sequence database has unique sequences that you may not necessarily find on NCBI website, especially the sequences that come from patents and certain sequences that needs to be manually curated from the full text. Okay, so for those who are doing sequence search, you might want to give it a try because you do get to search NCBI sequences anyway. So now you get some additional content, sequence content from CAS. Okay, so this is all I have. It's a very quick introduction, and actually we have 2 other databases here, which maybe we could, do this training for the 2 other databases at another time. Okay, so I'll hand things over for, I think, is it Q&A?
Thank you, Pam. Yeah, we have a few questions here, but I managed to answer some of them. But I think there's one more question, which is I think someone is asking, like Fitri is asking whether we can actually search substance using mass spectra. So yeah, so unfortunately, I think we can't do that. But is there any other way that we can work around it?
For mass spectra, no, but you could search compounds by its mass. So like, for example, if you have a large, okay, let me just pull back. Okay, so we have a large set of substance data like this.
You're not showing your screen.
Oh, sorry. Okay, so let me share my screen again. Okay, if you have a large set of compounds like this, we do have a molecular weight, but I know this is not exactly the same as kind of the mass to charge ratio that you're probably more interested in, but this will be the closest in terms of trying to find compounds by mass and matching it with the mass that you see on the mass spec. Okay, I think we have this experimental spectra. So this will tell you which compounds have mass spec available in our SciFinder end. All right, but you can't actually search it by mass. So it depends what you are looking for. If you're just trying to find compounds with mass spectra, you have to first run a structure search, and then you could use the experimental spectra availability filter, okay, to find the compounds. But if you're more interested to go by the mass of the compound, then the closest you can go is the molecular weight, but not the mass to charge ratio.
Okay, thank you. I think Pak Yudi have a question. Asking how to search literature about polymer. Can we do it by drawing the monomer dimer?
Right. Yeah.
Then
okay.
SciFinder give the correct information. Yeah.
Yeah. So for polymer, it's actually quite a complex topic on its own. But to kind of summarize it, if you want to search polymers, yes, you're right. You could do it by the structure of the monomers. Okay, so I'll give an example. First, I'll search it by name. So of course, searching by name is the other option. Okay, I'm just using a common polymer, polyethylene terephthalate. Most of the polymers in SciFinder N, they are represented by monomers, all right, like what we see here. Monomers, but there are some polymers that actually have the structural repeating unit representation. So polyethylene terephthalate has structural repeating unit and the monomeric version. So both are structural searchable. So you could try both ways, but I would say majority of the polymers only have the monomeric representation. So if you want to search this by structure, this is how you would draw it. Okay, so I'm just going to add this into the editor. So you essentially just draw out the structures of your monomers. If you have 2, you have 3, you have 4, you just draw them out individually and just run the search, right? It doesn't matter that it's actually separate molecules. So that's one approach. The other approach is actually to use the component registry number search. So this is if you already have specific monomers in mind, right? And maybe you want to, and you know the registry number of the monomers. So let me open this up in a new tab. Okay, so you can see that for polymers, they have their own registry number, but for the individual monomers, the monomers have its own registry number as well. So in this case, these are what we call component registry numbers because it's just one component of the entire polymer. So when it comes to substance searching, right, we have the registry number as well as the component registry number. So the other approach you can try is to actually put in the component registry numbers. And if you have more than one component, you just have to add a separate row. Okay, and then you can run the search. So of course, you may pull back mixtures. So this, you can see, it finds us polymers that contain these 2 monomers with a third component. If you're not interested in the third component, you can always go to the number of components and just limit it to 2. All right, but you can get quite interesting results because you can find polymers with a third component, fourth component, etc. So this is the other approach, using the component registry number. And the component registry number is actually very interesting because now you can search polymers that contain cellulose or chitosan, right? Because those have no structures, but you can now search them with the CAS registry number. So let me just do a quick demonstration for that. First, let's see, I'm interested in cellulose. Right, no structure, so I can't search this. If I want to find polymers that contain cellulose and other monomers, I can't search this by structure, but I can actually search it using the component registry number. Okay, so I paste in the registry number of cellulose. Run the search. I could then use the substance class, okay, limited to polymers, because you may get other types of compounds as well. Okay, and then once you do that, you'll be able to find, did it apply? Yeah, okay, you'll be able to find polymers where one of the components is cellulose, okay, using the component registry number search. So a few ways to go about it. You can search it by name, search it by the structure of the monomers, or you can even search it by the component registry numbers of the monomers. Okay.
Thank you so much, Ms. Pamela. I have another question. What about peptide?
Peptide. Okay, so
Yes, thank you.
peptide. So peptide, I think the easiest way is actually to do blast search, because right. So so for this blast search, we like the earlier example. Okay, let me just edit in. So this is actually a peptide sequence, right? I'm doing it using the code, the amino acid code, which is a lot easier than drawing out the structure. Okay, so do a blast search. If you want something more specific, you can consider the motif search, because the blast search will find not only the identical sequence, right, but it tries to match it with larger sequences, and it also allows misalignments. So if you're not interested in that type of answers, you could go directly with a motif search. So let me copy this over here. So to do a motif search, it will allow you to find just exactly this sequence, but you need to put in some symbols. Okay, so the caret symbol and the $sign. So if you put in these symbols, then the system knows that I only want to find this exact sequence. Don't give me something that is longer or shorter. Don't give me any sequences that have some misalignments at certain positions. Then you could go ahead and run the search. And the reason why I say this is also because for peptide sequences, once it gets quite large, you don't actually have you don't actually see structural information in SciFinder, and so you can't search by structure. Okay, so let me see if I can think of an example. Like if I use the antibodies, right? Like trastuzumab. Okay, so these are over 1,000 long peptide sequence. Of course, there's the heavy chain, light chain. So you can see there's no structural information. So it's not possible to draw out the structure. So to find this type of sequences, peptide sequences, of course, if there's a name, that's the easiest. The other way would be to use the sequence search option that you see on our homepage earlier on. Okay, for shorter sequences, yes, you can try searching it by structure for peptides. Okay, but it's quite challenging to draw peptide sequences in SciFinder. All right, so we do have, I think we have the templates for the amino acids. So if you want to search it by structure, you can, right? Use the templates and then connect the amino acids manually. But I believe that's quite tedious. So probably the sequence search option is the best. Okay, so using this over here. Let me see if, did my search not register in the history? Let me see. Yeah, okay, it's still searching. So it does take a while for the results to be done. Okay, so hopefully that answers your question.
Thank you so much. Thank you so much.
No problem. Are there any other?
Yes, Pam. I think Fitri got another question.
Yeah, about the edit structure, right?
Yes.
Let's
Or maybe
say
you can
adding.
show.
Yeah, I'm reading the question.
Okay.
We can edit the chemical structure, yes. Adding component based on the findings, yes, exactly. You're exactly right. So so there was a question regarding the edit structure function, which is really a very useful feature. So let me again pull back. Let's see one of the search results. Okay, let's reuse this. And it could be, something even more complicated than this structure. So you could find like a similar or, the closest compound to what you are working with. And then exactly like what Fitri said, if you click on the structure image, all right, you can select edit structure. It transfers it into our draw editor. And then now you can make all sorts of modifications, right? Like you say, based on your NMR, maybe you notice that, oh, okay, there is a, isopropyl group somewhere. I can edit in here. And then you can rerun the search. And in fact, you don't even need to edit directly to the molecule. You can even do like fragment searches. Okay, so like, there's an isopropyl, but maybe you don't know, is it this oxygen, or this oxygen? Can leave it open like this. But of course, when you leave it open, you are opening up to more substitutions and more variations. So the other thing you can consider doing is blocking some of the positions from further substitution. So if you are pretty sure you want an isopropyl group, okay, then we can use the this lock atom. So we can block this position so it doesn't try to build off these carbons in my isopropyl fragment. I can even block this, and I can block all these. Let's say, right, if you are pretty sure that it shouldn't be on any of these positions, and we can leave the oxygen open. We have to leave this open, unfortunately, because potentially it needs to attach to one of the oxygens. So we can do what we call like fragment searches, right? You have different functional groups that is part of the molecule, but you don't know exactly where it's connected, and you want to check in the literature, right? What are some of the maybe known compounds that has this functional group and how they are attached? So I could try running this search and see if we get anything. Okay, so all right, so for example, this, okay, actually this is isobutyl, so I should probably have deleted this carbon. All right, but you can see what I mean. The fragment is attached to various parts of the molecule, and it doesn't even need to be directly attached. It could be via, some other connecting groups. Okay, they're not not that many, but yeah, we have some. Okay, so yeah, that's for the edit structure feature. Okay, let me see if there's anything else. Yes, you're absolutely right. It's really great for exploring new compounds. Yeah, so there was a question regarding the reaction protocols, right? So even if it's not published in English, if CAS indexes that article for synthetic protocols, you will be able to see the protocols in SciFinder N in English. Okay, but of course we don't include the protocols of every single journal published in the world. So that's something to be mindful of, which is why I mentioned that you should use the experimental protocols filter. Okay, so maybe let me kind of show that feature again, because I was kind of going a little bit fast for the reaction portion. So right over here, let's rerun this search. And it's the same, especially for formulations. Okay, a lot of the formulation data are actually found in pattern literature, and it could be a Japanese pattern, Korean pattern, even if you have access to the full text, right? So what if you don't understand that language? But for the formulations, we also translate it into English. Okay, so I'll switch to a larger answer set. So like I say, not every reaction comes with the protocol, like the ones here don't have the protocols, but fortunately there is the pattern, so you can access it directly from the pattern, except this is probably in Korean. So in this case, this won't be translated, okay, because we are going directly to the full text to get the pattern. But use the experimental protocols filter to find out which reactions come with the protocol that you can view directly in SciFinder N. So, and it will be in English. Okay, but in this case, the exact matches do not have protocol. So this is the other thing to take note of. So like, for example, this, you can click on it and view the protocol in English. Okay, let's see if there are any other questions.
Yeah, Pam, I think that's all.
Okay.
Yeah, thank you so much.
All right.
So we'll be handing over to Pak Indra or Pak Wandi.
Yeah, okay. If there's any more question, you can contact Pamela or Adrian in the chat box. There's their email. And if there's no question, I think I will give it back to the MC. Pak Indra, silakan Pak.
Ya, baik. Bagaimana Bapak dan Ibu? Apa masih ada pertanyaan lagi? Baik, jika tidak ada, mungkin kami akhiri untuk acara pada sore hari ini. Terima kasih Bapak dan Ibu atas partisipasinya dalam kegiatan kali ini. Kami selaku penyelenggara acara memohon maaf apabila terdapat kekurangan-kurangan dalam kegiatan pada saat ini. Kami tutup acara pada sore hari ini dan terima kasih. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. Selamat sore Bapak dan Ibu.
Thank you very much. Thank you very much for Ms. Pamela and Adrian.
Thank you very much. Terima kasih.
Thank you.
Thank you.